Well, thank you so much for this opportunity. It's my great pleasure to be here. So I wanted to begin with a little background about my evolution in my career from the benchtop to the laptop. And so I started off my graduate work in cellular immunology, studying macrophages in the context of mycobacterial infection and leprosy with Robert Maudlin, and I found the divergence of phagocytic and antimicrobial programs, which actually help cause the pathogenesis of leprosy. For my postdoc, I next dabbled in bioinformatics, looking at blood transcriptomes from TB patients. And analyzing the latent TB patients, I found that a host defense network and found that IL-32 at the center of this network was a correlation of protection in latent TB, and that IL-32 itself can kill MTB in infected macrophages. And so now I'm on a NIAMS K01 level grant and incorporating more translational medicine through this training program, as well as more computational experience by working with Matteo Pellegrini at UCLA. And so I'm trying to carve out a research niche in terms of translational computational immunology. And what you'll see is actually my career has come full circle, and I'm back to cellular immunology. And so one of the problems that I'm trying to address is the analysis of clinical samples, such as blood or patient biopsies, and the transcriptome or methylome analysis. And normally, this is a straightforward approach of averaging the cases of controls, find differential expressed genes, pathway enrichment, maybe find diagnostic proteins for diagnosis. But one of the problems is that these patient biopsies and clinical samples have differing numbers of cell types, which may confound the analysis of this data. And what we really want is these immune pathways. And so the question is how to account for this cell heterogeneity and individual patient variation, as we can also see. And so working with a graduate student, we developed a technique to dissect in silico the cell type and immune activation pathways within these samples. And so the basis of this is we basically extract immune cell type activation gene signatures from reference profiles from cell types and activation signatures, and then measure those within the sample transcriptomes. And what we get out is an individual score for each signature for each patient. So we get individual level patient information about their immune status. And I've used external validation in vitro and correlated that this correlates with the actual cell frequency down to 1 to 5%. I have also developed a method similar, actually using DNA methylation, but I don't have time to show that today. So kind of to give you a flavor of what this looks like, so working actually with Jeffrey Ginsberg, my mentor here at Keystone, and collaboration that he fostered with Micah McLean at Duke, was looking at immune monitoring of acute infection. And so so what happened was we had patients infected with influenza and then followed over the course of a few days for their blood transcriptomes. Input this into Savant, and what we could see is that in the beginning we see a decrease in the neutrophils leaving the periphery, presumably for the lung, followed by an increase in the type 1 interferon response, a known antiviral response, as well as a decrease or a leaving of the CD8s from the periphery, which may be involved in memory. And so to look at more chronic disease, working with an undergraduate, Leah Briscoe, we looked at TB blood transcriptomes, over 700 from 4 different cohorts. A lot of these cohorts are actually from Ann O'Gara's lab here, and I hope you don't mind me using a lot of your data. I would love to hear your input in this. And so we input this into the Savant tool. We get a heat map here of different immune signatures, and I'm just going to highlight a couple of them here with the CD8 and type 1 interferon. But overall, what we could see is that each one of these is from one cohort in one condition. These are from active TB patients, latent TB patients, other respiratory diseases, and with and without HIV co-infection. What we could see from all these different data sets, different labs, we see them clustered by their immune their immune conditions, such as active TBs here on the right, latent TBs and treated TBs on the left, and separated out by their HIV immune response. So we could see in detail a large amount of information. And so what we wanted to try here is, well, can we classify TB based on the cell signatures alone? And what we did, we found that with latent versus active versus healthy, we get about 75% accuracy. And so looking just more directly at flow cytometry, getting some data from Subhas Babu at the NIH, we're actually able to get 90% accuracy or over 90% accuracy to diagnose TB using just cell signatures alone. So we don't have to actually do a blood transcriptome from every single patient. We could just look at their cell frequency. And so with that, I would like to acknowledge the Keystone Fellows Program, especially Arlene has been wonderful and outstanding in leading this effort, as well as all the ASB members for tolerating us and being actually very quite welcoming to all the fellows here. And my mentors, Matteo Pellegrini, as well as Jeffrey Ginsberg. I think the top-down approach from the Keystone Fellows Program is really unrivaled by any other diversity program that I've seen, and the focus on diversity I'm really amazed by with Keystone. And with that, I just want to give a quick plug that I'm looking for collaborations. If you're looking for looking at immune monitoring in your particular system, either RNA transcriptomes or DNA methylomes. Plus, I'm also looking for faculty positions this fall. Thank you.
Okay. I thought your presentation was really, really exciting and very interesting. And my question is, would you have done this work with Dr. Ginsberg had you not been a fellow at Keystone? Would this have happened for you?
No, definitely not. He's been very outstanding because he's basically the type of researcher that I would like to be in terms of a personal medicine approach to genomics research. And that's why I kind of sought him out as a mentor. And he's been very helpful in terms of giving me honest, real feedback, not just a pat on the back, and actually fostering this collaboration with one of his other faculty there, Micah McLean. And so I think that's been really a great and hopefully a boon to my research going forward.
Well, it's very impressive work. Congratulations.
Thank you.